Summary of precise RNA gene editing technology. Key developments in gene editing technology. The field of precise RNA gene editing technologies has seen remarkable advancements since the discovery of DNA's double helix structure in the 1950s. These innovations have led to various methods for targeted genome modification, significantly impacting research in genetics, medicine, and biotechnology. 1. Timeline of key events. 1950s, the discovery of DNA's structure provided the foundation for genetic manipulation. 1960s to 1980s, introduction of restriction enzymes and the development of homologous recombination techniques. 1990s, emergence of zinc finger nucleases ZFNs and transcription activator-like effector nucleases TALENs for targeted DNA editing. 2012, the CRISPR/Cas9 system was introduced, revolutionizing genome editing with its simplicity and efficiency. 2016, development of base editing and prime editing technologies allowing for precise nucleotide changes without double-strand breaks. 2. Gene editing technologies. CRISPR/Cas9, this system employs a guide RNA to direct the Cas9 nuclease for precise DNA cuts, enabling simultaneous editing of multiple genes. However, it presents challenges such as off-target effects. Base editing (BES), this method allows for the conversion of one base pair to another without generating double-strand breaks, although it is limited to specific transitions. Prime editing (PES), combining a Cas9 nickase with reverse transcriptase (PES) facilitate a broader range of edits, including insertions and deletions with lower off-target effects compared to traditional methods. Click editors. This recent technology integrates HUH endonucleases with DNA-dependent DNA polymerases in CRISPR-Cas9 nicases to allow programmable genome engineering. 3. Applications. Gene therapy. Precision editing can correct mutations linked to genetic diseases employing techniques like HDR homology-directed repair and high-tie homology-independent targeted integration. Disease models. Gene editing is crucial for creating animal models that replicate human diseases. Aiding research and treatment strategies. Functional genomics. CRISPR-based screens enable the generation of genetic variants for studying gene functions. 4. Challenges and limitations. Delivery systems. Efficiently delivering gene editing components remains a significant challenge, with various methods like plasmids, viral vectors e.g. AAV, and ribonucleoprotein RNP complexes being explored. Editing efficiency. Despite advancements, achieving high HDR efficiency remains a barrier for therapeutic applications. Off-target effects. Off-target editing poses risks, particularly with CRISPR/Cas9 systems. Strategies to mitigate this include using modified Cas9 variants and optimizing sgRNA designs. 5. Future directions. Ongoing research aims to enhance the efficiency and specificity of gene editing tools. The development of new Cas variants and improved delivery mechanisms remains a key focus. Integrating precise editing technologies into clinical settings holds transformative potential for treating genetic disorders, with several approaches advancing to clinical trials. In conclusion, the landscape of precise RNA gene editing technology is rapidly evolving, marked by innovative methodologies and diverse applications across biological fields. While challenges persist, the future holds promise for significant advancements in therapeutic interventions and functional genomics. Key sources and references. 1. Nature Biotechnology. Recent advancements in precise genome editing, including the development of click editors and their implications for programmable genome engineering. URL: Nature Biotechnology www.nature.com/articles/S41587-024-02340X. 2. Research articles. Comprehensive reviews discussing the mechanisms, strategies, and applications of precise genome editing in human diseases, highlighting the evolution from early gene editing techniques to modern CRISPR/Cas9 and its derivatives. URL: Signal Transduction and Targeted Therapies www.nature.com/articles/S41392-024-01750-2. 3. PubMed and other journals, numerous studies detailing the practical applications of gene editing efficiency improvements and future challenges in the field. URL: PubMed/pubmed.nlm.nih.gov/386565525/. These references provide a comprehensive overview of the current state and future prospects of precise RNA gene editing technologies, emphasizing their significance in modern genetics and medicine. The content provided delves into the advancements and intricacies of precise RNA gene editing technologies, highlighting key developments, methodologies, applications, and challenges faced in the field. Here are some counterintuitive insights derived from the provided information. 1. Perception of CRISPR's simplicity versus actual complexity. While CRISPR/Cas9 is often celebrated for its simplicity and ease of use in genome editing, the reality is that achieving precise edits without off-target effects is a complex challenge. Many researchers initially assumed that CRISPR's straightforward design would inherently lead to high accuracy. But they have since discovered that off-target modifications can complicate outcomes significantly. 2. Base editing limitations. Base editing technologies are often viewed as a highly precise tool that can introduce specific point mutations without double-strand breaks. However, despite their efficiency, they are limited in their ability to perform certain types of edits such as transversions or insertions and deletions, which means that researchers must still rely on other methods for a broader range of applications. 3. Homology-directed repair HDR efficiency. There's a common belief that HDR is a universally efficient method for repairing double-strand breaks and facilitating precise genome modifications. However, HDR is often limited to dividing cells and has a low efficiency rate, particularly in non-dividing cells. This contradicts the expectation that HDR could be a straightforward solution for all gene editing needs. 4. Role of non-homologous end-joining NHEJ. Often considered a less desirable repair pathway due to its propensity to introduce errors, actually plays a critical role in the efficiency of certain editing strategies like HiTi. This highlights a counterintuitive aspect where a pathway typically viewed as problematic can provide essential support for targeted integrations and editing in certain cellular contexts. 5. Off-target effects of advanced technologies. There is a perception that newer technologies like prime editing and base editing are inherently safer and more precise than CRISPR/Cas9. However, studies have shown that these advanced methods can still produce off-target effects, indicating that no editing technology is without risk. This challenges the assumption that newer techniques are automatically superior in terms of specificity. 6. Clinical application of gene editing. Many assume that once an editing technique is developed in the lab, it can be rapidly translated into clinical therapies. However, the transition from laboratory research to clinical application is fraught with regulatory, ethical, and technical challenges that can significantly delay the availability of these therapies. 7. Delivery mechanisms' impact on efficacy. The expectation might be that any delivery system, viral vectors, RNPs, etc., can efficiently deliver genome editing components to all cell types. In reality, the choice of delivery system can dramatically affect editing efficiency and specificity, often requiring extensive optimization for different cell types and tissues. 8. The role of cell cycle in editing efficiency. It is widely believed that gene editing is most effective during certain phases of the cell cycle, e.g. S/G2 phases for HDR. However, recent findings suggest that HDR can also occur in postmitotic cells under specific conditions, which contrasts with the traditional understanding of cell cycle dependency in genetic modifications. These insights reveal the nuanced realities of precise RNA gene editing technologies, emphasizing that assumptions made based on initial observations or generalizations may not always hold true when faced with the complexities of biological systems. The report on precise RNA gene-editing technology outlines significant advances in gene-editing methodologies focusing on CRISPR/Cas9 base editing, base prime editing, Ps, and newly developed click editors. While key advancements and challenges are summarized, several significant aspects may be overlooked by many readers which are crucial for understanding the technology's implications in future directions. Overlooked aspects and their significance. 1. Precision versus efficiency trade-offs. Why it's overlooked. Many discussions about gene-editing technologies emphasize their precision in specific applications without delving deeply into the trade-offs between precision and efficiency in different contexts. Significance. Understanding that while prime editing offers high precision, it often does so at the cost of lower efficiency compared to methods like CRISPR/Cas9. This is critical for therapeutic applications where high editing rates are necessary. For example, in clinical settings, achieving a high percentage of edited cells can be more important than achieving perfect precision, especially when targeting large populations of cells in vivo. 2. Off-target effects and mitigation strategies. Why it's overlooked. The report mentions off-target effects associated with CRISPR/Cas9 but does not extensively discuss newer strategies to mitigate this issue, such as the development of modified Cas9 variants and optimized guide RNA designs. Significance. The ongoing development of Cas9 variants that reduce off-target effects is essential for enhancing the safety profiles of gene editing applications in humans. As gene therapies move into clinical trials, these improvements can make a significant difference in patient safety and efficacy. 3. Delivery mechanisms. Why it's overlooked. The efficiency of gene-editing technologies often overshadows the challenges related to delivering these tools into target cells or tissues. Significance. Delivery is a critical barrier that influences the success of gene-editing therapies. Innovations such as lipid nanoparticles LNPs and virus-like particles VLPs for delivering CRISPR components are vital for improving the effectiveness of treatments. Recognizing the importance of delivery can aid in addressing practical challenges in applying these technologies in therapeutic settings. 4. Clinical translation and regulatory hurdles. Why it's overlooked. There is often a focus on the scientific advancements without addressing the regulatory landscape that influences the translation of these technologies to clinical use. Significance. Understanding the regulatory challenges and the need for thorough clinical testing is essential for the adoption of gene editing in medicine. Awareness of these hurdles can help researchers and investors align their efforts with regulatory requirements, facilitating smoother transitions from lab to clinic. 5. Ethical considerations. Why it's overlooked. The report primarily emphasizes technical advancements, often neglecting the ethical implications of gene editing, especially in human germline editing. Significance. Ethical considerations are paramount as gene editing technologies progress. Discussions about potential misuse, long-term impacts on the human gene pool, and societal implications must accompany technological advancements. This awareness is crucial for guiding responsible research and public policy. 6. Role of RNA in genome editing. Why it's overlooked. While the report mentions RNA components in gene editing, the broader implications of RNA's role in gene regulation and the potential for RNA-targeted therapies may not receive adequate attention. Significance. RNA's involvement in gene expression regulation presents opportunities for new therapeutic approaches such as RNA interference RNAi and antisense oligonucleotides. Recognizing the potential of RNA technologies can open up additional avenues for treatment strategies, particularly for diseases where traditional DNA editing may not be effective. 7. Emerging technologies in future directions. Why it's overlooked. The focus on established technologies like CRISPR/Cas9 base editing and prime editing may overshadow emerging methodologies such as click editors and other novel systems. Significance. Emerging technologies may offer solutions to current limitations in precision and efficiency, expanding the toolkit available for genome editing. Staying informed about these advancements is essential for researchers aiming to remain at the forefront of biotechnology. Conclusion. The report provides a comprehensive overview of precise RNA gene editing technologies, highlighting key advancements and challenges. However, the significance of aspects such as the balance between precision and efficiency, off-target effects, delivery mechanisms, regulatory hurdles, ethical considerations, the role of RNA and emerging technologies is crucial for fully grasping the landscape of gene editing. Addressing these points can foster a more holistic understanding of the potential and implications of precise RNA gene editing technologies, guiding both research and application in real-world contexts. 1. Technological advancements and mechanisms. The landscape of RNA gene editing technology has undergone dramatic transformations over the past few decades, evolving from rudimentary methods to sophisticated techniques. The introduction of CRISPR/Cas9 in 2012 marked a pivotal shift in the field, allowing for precise genome modifications with ease and efficiency. Following this, innovations such as base editing and prime editing have emerged, enabling targeted nucleotide alterations without inducing double-strand breaks. Base editing Bs allows for conversion of specific base pairs without DSBs, while prime editing Ps combines the Cas9 nickase with reverse transcriptase to facilitate a broader range of edits. The development of click editors, which integrate various components for programmable genome engineering, showcases the continuous evolution of these technologies. These advancements not only enhance precision and efficiency but also expand the range of possible genetic modifications, paving the way for novel therapeutic applications. 2. Applications in gene therapy and disease models. Precise RNA gene editing technologies have significant implications for gene therapy and the development of disease models. By allowing for the correction of mutations associated with genetic diseases, these technologies hold the potential to transform the treatment landscape for conditions like sickle cell disease, beta thalassemia, and various cancers. The ability to create accurate animal models of human diseases through precise genome editing aids researchers in understanding disease mechanisms and testing new therapeutic strategies. For example, CRISPR/Cas9 has been utilized to generate mouse models mimicking human conditions, facilitating research into targeted therapies. This intersection of gene editing and therapeutic application underscores the importance of precise RNA gene editing technologies in advancing biomedical research. 3. Challenges and limitations. Despite the promising advancements in RNA gene editing technologies, several challenges remain that hinder their widespread application. Issues related to delivery systems pose a significant barrier, as efficient delivery of gene editing components to target cells is critical for successful outcomes. Various methods, including plasmids, viral vectors, and ribonucleoprotein RNP complexes, have been explored, but each comes with its own set of limitations. For instance, the size constraints of adeno-associated viral vectors restrict the packaging of larger editing components. Furthermore, the efficiency of editing remains a concern, particularly for methodologies like homology-directed repair (HDR), which is often limited to specific phases of the cell cycle. Off-target effects also present challenges, particularly with CRISPR/Cas9 systems, necessitating ongoing research into optimization strategies that enhance specificity and reduce unintended edits. 4. Future directions and innovations. The future of precise RNA gene editing technologies is poised for continued innovation, with a focus on enhancing efficiency, specificity, and delivery mechanisms. Research into new Cas variants with improved targeting capabilities and reduced off-target effects is underway, alongside efforts to develop more effective delivery systems tailored to various cell types and tissues. The integration of these technologies into clinical settings holds promise for revolutionary advancements in treating genetic disorders. Moreover, the potential for combining precise editing tools with other therapeutic modalities could yield even more effective treatment strategies. The ongoing exploration of novel applications, including molecular recording and programmable gene therapies, reflects the dynamic nature of this field and its capacity to impact diverse areas of medicine and biological research. Here are three famous historical cases that are similar in nature to the advancements and implications of precise RNA gene editing technology. 1. The Human Genome Project (HGP). Launched in 1990 and completed in 2003, the Human Genome Project was an international scientific research initiative aimed at mapping and understanding all the genes of the human species. This monumental effort involved sequencing the entire human genome, which comprises about 3 billion DNA base pairs. The HGP provided the foundation for numerous advances in genetic research, including the identification of genes associated with diseases and the development of new diagnostic and therapeutic strategies. It paved the way for subsequent technologies, including gene editing. By enhancing our understanding of genetic variation and its implications for health and disease. 2. CRISPR-Cas9 technology. While not a historical case in the classical sense, the development and commercialization of CRISPR-Cas9 technology in the early 2010s represents a revolutionary moment in gene editing history. Initially discovered as a bacterial immune system, CRISPR-Cas9 was adapted for precise gene editing in various organisms, including humans. The technology enables researchers to make specific alterations to DNA sequences with unprecedented ease and efficiency. It has vast applications in medicine, agriculture, and biotechnology, leading to potential cures for genetic diseases, improvements in crop resilience, and the creation of genetically modified organisms. The ethical implications and potential for misuse of CRISPR technology have spurred significant discourse, similar to the discussions that arose following the mapping of the human genome. 3. Zinc finger nucleases (ZFNs). Developed in the late 1990s and early 2000s. Zinc finger nucleases were one of the first engineered gene editing tools used to create double-strand breaks in DNA at specific locations. This technology allowed for targeted modifications of the genome and laid the groundwork for later advancements such as TALENs and CRISPR. ZFNs were utilized in early gene therapy trials and research, demonstrating the potential for gene editing technologies to treat genetic disorders. Although ZFNs have been largely superseded by more efficient technologies like CRISPR, they mark a significant step in the evolution of genetic engineering and share similarities in their precision and application in therapeutic contexts. These cases highlight the evolution of genetic manipulation technologies and their profound implications for science, medicine, and ethics. Paralleling the advancements seen in precise RNA gene editing technologies today. Future outlook for precise RNA gene editing technology. 1. Advancements in editing techniques. The field of precise RNA gene editing is poised for significant advancements in the coming years. With ongoing research and development, we can expect improvements in the efficiency and specificity of existing methodologies such as CRISPR base editing and prime editing. The introduction of novel systems such as click editors, which utilize a combination of HUH endonucleases and DNA-dependent DNA polymerases, holds the promise of enabling even greater precision in genome modifications. These advancements will facilitate the development of versatile tools that can operate across a wider range of genetic targets. 2. Enhanced delivery mechanisms. One of the significant challenges in gene editing remains the effective delivery of editing components into target cells. Future innovations are likely to focus on developing advanced delivery systems, such as lipid nanoparticles and virus-like particles, which can improve the stability and bioavailability of gene editing reagents. These systems will enhance the capacity to deliver multiple components simultaneously, thereby increasing the efficiency of gene editing in vivo. Additionally, personalized delivery methods that consider the specific requirements of different tissues and diseases may lead to more effective therapeutic applications. 3. Broader therapeutic applications. As precise RNA gene editing technologies become more refined, their applications will expand beyond traditional genetic disorders. Future therapies could target complex diseases such as cancer, heart disease, and neurodegenerative conditions by correcting mutations, modifying gene expression, or introducing novel genes. The potential for gene editing to create tailored treatments for individual patients, often referred to as personalized medicine, is on the horizon, which could drastically change how we approach disease management and therapy. 4. Regulatory and ethical considerations. With the rapid development of gene editing technologies, regulatory frameworks will need to evolve to address the ethical implications of such advancements. Future discussions will likely focus on the safety and long-term effects of gene editing in humans, especially concerning germline editing. Establishing comprehensive guidelines will be essential to ensure that gene editing therapies are both safe and ethically sound, fostering public trust and acceptance. 5. Integration with other technologies. The future of precise RNA gene editing will likely see an integration of various technologies, including artificial intelligence (AI) and machine learning, to optimize gene editing outcomes. AI can assist in designing more effective guide RNAs, predicting off-target effects, and analyzing large datasets generated from genomic studies. This interdisciplinary approach may lead to groundbreaking advancements in gene editing precision, efficiency, and application. 6. Development of robust disease models. The creation of accurate disease models will become increasingly important as gene editing technologies advance. Future research will likely focus on developing sophisticated animal models and cellular systems that mimic human diseases more accurately. These models will be essential for testing the efficacy of new gene editing therapies and for understanding the underlying mechanisms of diseases. 7. Educational and public engagement efforts. As gene editing technologies continue to advance, there will be a growing need for public education and engagement to address misconceptions and promote understanding of the science behind these technologies. Outreach programs and educational initiatives will be crucial in fostering informed discussions on the benefits, risks, and ethical considerations associated with gene editing. Conclusion. The future of precise RNA gene editing technology is bright, with potential advancements that will enhance the precision, efficiency, and applicability of gene editing in medicine and research. Ongoing improvements in editing techniques, delivery mechanisms, and regulatory frameworks will pave the way for exciting therapeutic possibilities. Through interdisciplinary collaboration and public engagement, the field can navigate the challenges ahead while maximizing the benefits of these transformative technologies.